Okay, so welcome back after lunch break. So the next speaker is Neda Barghi. She is in Vienna, in the POPGEN, will be basically relocating middle of this year to Plön in Germany. And thank you.
Okay, good afternoon, everyone. My name is Neda, and today I'm going to tell you about how we can study polygenic adaptation in evolution experiments in Drosophila. So I think I wasn't here, but I think the past few lectures, since in the past 10 days, you've already got a really good understanding of the basics of adaptation with very good lectures. So that is why today I will just briefly go through a few introduction or background just to tell you what I think is polygenic adaptation. And then I'm going to talk about why I think evolutionary experiments are a really good approach to understand adaptation. And then I'm going to show you a majority of unpublished data about experiments that allows us to study the patterns of adaptation as populations evolve to a new optimal phenotype, and also the effect of population size or drift on these adaptive processes, and also the effect of selection pressure, or as Luis talked about it, a distance to the nutrient optimum on these adaptive processes. So I think there's no need to mention to this audience that majority of quantitative traits and complex traits are polygenic with a lot of contributing loci. We've already seen genetic basis of human size, human height, that even after 5 million individuals being sequenced, still we can explain only around 45% of variance, and also type 2 diabetes, and of course thermal adaptation also in Drosophila. So we already know that genetic basis of these traits is polygenic, but how does adaptation of these polygenic traits work? So relying on what Luis mentioned, majority of complex traits are under stabilizing selection, which basically stabilizes populations mean around a mean value, which is known as a trait optimum. And upon some environmental change, a population experiences a novel selection pressure on a trait that has a polygenic basis. So population has quickly shifts and evolves phenotypically to meet this new optimum. But what is the genetic basis of this phenotypic change? So I think in the past 6 or 7 years, I've been hearing that there are two schools of thoughts. Either adaptation occurs through large frequency changes at independent alleles, and this leaves a very clear fingerprint, which has been used in many tests of adaptation, which is mainly selected sweep, hard or soft, complete or incomplete. And you could also think of it as a from the quantitative trait, quantitative genetic perspective, where adaptation occurs through small shifts at very many loci. And this normally leaves not very prominent signature at the genomic level, and therefore identification of it is more difficult. But I think it has been clear after Nick's talk that these two, basically large allele frequency changes and small shifts, are the two ends of the same spectrum. And I think we're going to hear from Joachim tomorrow that the results show that adaptation can actually occur throughout this spectrum. So in one population, depending on the redundancy or mutational target size, different individuals can use the same, if the mutational target size is small, different individuals can use the same beneficial allele, then that's why we do observe large frequency changes. Or if there are many adaptive alleles, different individuals in a replicate can use different alleles to reach the same phenotypic optimum, and then that's why we would have small shifts. So adaptation could actually occur anywhere along these two spectrums. And what I do find interesting and very fun about polygenic adaptation is the redundancy that also Sam talked about. Because if the number of beneficial alleles are more than what a population requires to reach this new optimal phenotype in this new environment, which we called genotypic redundancy, then we would expect low repeatability because depending on what is available in that particular environment, each population would use this handful of adaptive alleles and they can reach the same fitness optimum. And that would mean that we would have low repeatability. So in cartoon, I'm showing that the two replicates can converge phenotypically, and one replicant can use allele blue and green, and another one can use alleles red, purple, and orange to reach the same optimal phenotype. And I think that we've probably realized that there are a lot of approaches to understand adaptation. We need theoretical models. We also need empirical data as a basis of those models and also to test those models. And this empirical data could come from natural populations. There are a lot of clinal studies and also in Arabidopsis, in flies, in humans, and also evolution experiments. And I hope that by the end of this talk, you're convinced that evolution experiments could be really useful to provide good insights into adaptation of complex traits. So there are particular advantages of experimental evolution that I basically added the third one yesterday because I noticed this is also important. So first of all, what is important in evolution experiments is that we can have replication and we can see how adaptation occurs in multiple replicates independently in the same environment. And replication is a luxury that a lot of natural populations do not have. But also a possibility of collecting time series allele frequency and phenotypic data is also quite important, as we've also observed that there are particular methods that really rely on time series data. But also, experimental evolution allows adaptation of populations to a particular selection regime, which could be simpler or slightly more complex. But this selection regime can be constant throughout a very long period of time. So we could observe, I don't know, for example, stabilizing selection in these particular regimes. So when I joined Christian's lab as a postdoc, I started analyzing a particular data set where Christian has established some founder populations from Drosophila stimulans, and there were 10 independent replicate populations, all established from the same founder, and they were exposed to a new selection pressure, which was a hot selection environment. And this fluctuates between 18 degrees at night to 28 degrees during the daytime. And they were evolved independently, and we had time series data, genomic level, every 10th generation until generation 60. And flies are, I think even till now, are in the lab up to generation 300 or so. So we wanted to know what is the genetic basis of adaptation to temperature in these flies. So we initially started to phenotype these populations to see whether they have changed phenotypically after these 100 generations. So we measured fecundity as a fitness component. And so in the red ones are the evolved flies, and then the beige color is the ancestral population. So we observed that after 100 generations of adaptation in this environment, females are more fecund, they lay more eggs, they also produce more carbon dioxide, they also have significantly less lipid content, and these traits might be all also correlated. And the other thing that we notice is that there is no significant difference in these three traits among the 10 evolved replicates. So they've all converged phenotypically. And when we look at the genomic data, and sorry, I think you've probably seen this plot sometimes already. So
sorry, I had a quick, on the previous slide. So you measured fecundity. Did you measure something like sperm mobility in males?
No.
Just given the differential performance of males and females?
True, no, because I think measuring fitness, fecundity in females is easier. But yeah, that can also be done in males, but we have not really done it. But you mean the sperm content or mobility or whatever?
You can imagine a couple of ways of doing that. Like a lot of people study Drosophila, where you could do a sperm count on a microscope slide, or you could basically cross them back to a control female outside the group and compare their fertility relative to from the control.
Yeah, this could be, but no, we have not. Yeah. Yes. Yeah. No, we have not done it. Good suggestion. Okay. So at the genomic level, the take-home message is that there is a lot of heterogeneity among the 10 replicates. So this is one particular allele, and by allele I mean selected haplotype blocks, because we have a lot of linkage. So we cluster the SNPs based on the correlation in the allele frequency, and we call a block of selected alleles just basically a selected haplotype or selected allele. And you see that each box corresponds to allele frequency in one replicate, and in some replicates we have increase in frequency for this particular allele, and some of them we do not. To quantify this heterogeneity more systematically, we use summary statistic that we call a replicate frequency spectrum, and that is simply the frequency distribution of replicates depending on which or how many adaptive alleles increase in frequency in them, which basically means that 4 alleles increase only in one replicate, 8 alleles increase in 2 or 3 replicates, and so on and so forth. So you see that the majority of selected alleles that we have identified increase in frequency in 4 to 6 replicates. So there is some redundancy or heterogeneity in the selection response. And we wanted to see whether which evolutionary model would fit the heterogeneity that we observe in our empirical data better. So we performed a series of selection simulations. Some of them selected sweep, which basically each allele increases in frequency independent of other alleles. And we performed adaptation of a quantitative trait after a shift in trait optimum, which basically is equivalent of the model that Luis Miguel introduced as the one that undergoes a sudden shift, which basically gradually the selection coefficient decreases. And then for each of those rounds, simulations, we compute replicate frequency spectrum, which measures the heterogeneity among the replicates, and we compare it to our empirical data. So the lower it is, it basically means that it is closer to our empirical data, and then the higher it is, it basically means that they are more different. And this shows that a quantitative adaptation of a quantitative trait after a trait shift in trait optimum matches the heterogeneity that we observe in our data set much better than a selected sweep. So up to here, we saw that thermal adaptation in flies is polygenic. There are a lot of alleles, adaptive alleles that contribute to this. We also showed that we have genomic heterogeneity despite convergence at a phenotypic level. And we also saw that this redundancy actually is good because it facilitates adaptation. Each replicate could find a different evolutionary pathway to reach this new optimum in this new environment. So I think already you've heard me talk about fitness optimum or trait optimum several times, but it is very clear that in this data set and also in a lot of experimental and natural populations, we have no information about what the selected trait is or how far the new trait optimum is. But understanding when the trait optimum is reached, even though we do not know what a truly selected trait is quite actually important. And then the reason is that imagine a polygenic model where the trait or phenotype is basically mapped to fitness using a Gaussian fitness distribution, a fitness function that we already saw before. And then so in the initial phase of adaptation, when the after a sudden shift, the selection is mostly directional, and then we have large frequency changes. But after we have this, the new optimum is reached in this new environment, selection would be mostly stabilizing. So alleles initially undergo some drift, or basically they plateau, and later on, after a long period of time, they either get fixed or they are lost. So understanding this, dividing this phenotypic or the adaptation process into directional and stabilizing selection is important because it allows us to study stabilizing selection, the allelic dynamics under stabilizing selection, which may not really happen in nature because the environment might be constantly changing. And finding when the trait optimum is also reached is important because it allows us to identify adaptive alleles, because this is the phase that they have the most extreme frequency changes. So, so we did some simulations to show what are the features that we could use to identify polygenic adaptation, adaptation of a complex or quantitative trait after a shift in its trait optimum. And so it seems that patterns of phenotypic change could actually be a really good distinguishing factor to identify these phases. And then the reason is that in the beginning, after there's a shift in trait optimum, the phenotype changes until it reaches the new optimal phenotype in this new environment. And after that, the mean does not change, while the variance do change. And the difference, this pattern would be different if the population size actually is different. For example, in a small population size of, I don't know, 400 ME, which is shown here in this solid line, adaptation is less efficient, and it would take them a slightly more time to reach the optimum. But when the population is larger, it's going to be much more efficient or faster. So we decided to test this empirically. So we set up a founder population by using around 90 inbred lines. So these are all haplotype sequenced. And then we performed round-robin crossing, which basically you cross every pairs of lines. And we maintained them for around 15 generations to break the haplotype blocks as much as possible. And then we set up large and small populations. So our large populations are 6 replicates of around 100,000 each. And the small populations are around 20 replicates of around 800. And for those that are not really familiar with the fly work, this 100,000 is quite a large population size, because normally in the evolutionary experiments, we keep the populations around 1,000 to 2,000 individuals. And then as a new, basically, selection environment, we expose them to a high protein diet. So this new protein, or this new diet, it has, I think, twice more protein, and the amount of carbohydrate is also less than the ancestral diet.
Provide more resources to, I guess, to the larger ones, or?
We kept the space and food surface constant, basically.
Per individual or?
Per individual.
Okay.
Yeah. So the surface of the space and then the food is 125 times more in the large population than in small.
So how big is that?
It's 60 by 60 by 120 centimeter. But it's just, we do provide food in 2 layers, so we can keep more flies in it.
Thanks.
And since our hypothesis was that we would be able to observe a change in the phenotypic pattern as populations adapt to this new optimal phenotype, we had to collect time series phenotypic and genomic data. So collecting the genomic data is pretty easy. We collect it every 2 or 3 generations. But for the phenotypic part, it is more troublesome. So it is not very frequent, and also the time intervals are not really even. But yeah, I think it's still good. So we phenotype our populations at generation 0 before we expose them to any selection pressure, to any new high protein diet. And after 7 and 31 generations of adaptation, we also phenotype them at generation 60. But today I will show most of the data that I'm going to present belongs to this 31st generation of adaptation. So we will start with a high order phenotype, which is basically, yes.
Yeah. What is that phenotype versus?
Oh, this phenotype, the phenotype that we have gene expression data. We also have some measure some metabolites, but we also have some high order phenotypes like fecundity. So we try to collect them at multiple biological hierarchies.
So at time step generation 7, you have measured some phenotype. Is that what you meant?
Pardon?
I did not understand this.
So we phenotype flaws at generation 0, at generation 7, at generation 31. Yes. Any more questions? Yes. For gene expression, it's 40 flies, 50 males. And for our metabolite, it's just metabolomics, it's just metabolites, just 1 or 2. These are, I don't know, 8 or 9. So we'll first look at the fecundity, which is a measure of, as a proxy of fitness. So this one is the fecundity of the founder population before it is exposed to any selection pressure. And these are the fecundity of evolved populations. So we see that after 7 generations, we have a rapid change, significant change, increase in fecundity. But this level actually plateaus after generation 7. And also this generation 60, which is a little bit darker, is because this population, it is not large. Oh, first of all, this is about the phenotype in the large population. And because maintaining these large populations were quite strategically difficult. So after generation 40, each of those 6 replicates were bottlenecked. So we kept them in 3. Each big population is now converted into 3 small populations. And after 20 generations, we phenotype them. And we see that even after a bottleneck and 20 generations of adaptation, we do not observe any significant difference in the in fecundity. So that may suggest that adaptation is fast, and your new optimal phenotype, whatever that might be reached as early as 7 generations. And then I want to emphasize that this increase in fecundity is kind of like 2 different traits, because we increase for the higher reproduction, but also early fecundity. So here is the same data set, but the previous one was the sum of eggs that each fly lays during the course of 1 week. And now we just divide it into 7 days. So this is the founder population, generation 7 and 31. And we see that in addition to the increase in the number of eggs that these flies lay, so the peak of fecundity is also shifted towards earlier days, so day 1, and also at the generation 31, day 1 or 2 are the ones that basically have the peak of their fecundity. So it's kind of like adaptation for early fecundity and higher fitness, higher fecundity.
There's maybe a stronger signal here than you realize, because it's all early reproduction. So you can use some of these age-adjusted measures of fitness. And I think what you'll see is that the response is even more dramatic, because you've shifted a lot of the production onto early on.
Yes, because the flies that are 5 or 6 days old are normally used to establish the next generation. And that is why I think if you are more fecund at a younger age, it is more beneficial. And that's why we're selecting for early fecundity. You're right. More questions? Okay. And now we look at gene expression. And so here is a PC of founder population. Here is shown in orange, and the evolved population. So the light purple is a generation 7 of these large populations, and the purplish darker purple is at generation 31. So we see that PC1 separates our founder population from the evolved ones, and they all kind of cluster nicely together. And when we look at PC1 and PC3, then we could separate the generation 7 and 31. But this also kind of shows that phenotypically, or at the gene expression level, these 2 populations, the population 23 generations apart at generation 7 and 31, their gene expression is highly correlated. And you could see this higher correlation, maybe better in this plot. So I want to show you a couple of these plots. I'll walk you through this. So in the x-axis, we have log-2 fold change of genes at generation 7 compared to the founder population. And the y-axis is a log-2 fold change of genes at generation 31 compared to the founder population. So whatever you see in this, along this diagonal colored in this brown or beige, but these are the ones that are shared in these 2 time points, and the genes and the magnitude and direction of their expression. And those green ones are specific to a particular time point, either generation 7 or 31. But we see that majority of differentially expressed genes, around 76% of them, are shared between these two time points. And I think it kind of shows or suggests that phenotypically, these two generations are, flies in these two generations are very, very correlated, suggesting that adaptation might have occurred really early on. So I initially told you that we did this experiment to look for a plateau in mean, yes.
Perhaps I missed something, but do you know if they were correlated with the ancestral one or expression level? What if you put the ancestral instead of generation 7, you put generation 0?
It's a fold change, the fold change of
Okay,
generation
it's fold
7
change relative to 0.
to 0.
Okay,
yes.
sorry.
Yeah. And then it's the fold change of these two that is correlated around 0.9 or so. So we want to identify those phenotypes that after a change compared to the founder population have plateaued. So now we use transcriptomic data as a quantitative trait. So now we have 6,000 differentially expressed genes. So we could treat each of them as a quantitative trait. And we looked for, try to classify them depending on their co-expression network or pattern. We tried different tools, but at the end we came up with the idea that manually classifying them is actually much easier. So for identifying gene expression pattern that we call as plateau, the gene should be differentially expressed at generation 7 and 31 compared to the founder population, but there should be no differential expression between generation 7 and 31. So it should be plateaued, and there should be no change in the direction of fold change. And if this is the case, you would call it that it has plateaued. And when we look at around 6,000 differential expressed genes, we see that 4% of them, around 4,000 genes, they show this sign of pattern of plateau, meaning that after a significant increase or decrease in their expression, we do not observe any significant change afterwards. Again, this might suggest that majority of the genes, majority of these traits have plateaued after the initial adaptation. So, but we did identify 35% of genes that actually did not plateau. And when we look at them, we see that some of them, we call them monotonic, which basically they continuously increase or decrease their expression compared to the founder population. These are around 400 genes, and there are other genes that we call them late response, and we basically do not identify them as differential expressed at generation 7, but we do find them as differential expressed at generation 31. So I think they might be, we were interested to see what are these genes that they are still continuously changing in their expression. So we did go enrichment analysis, and we found a lot of Go terms related to lipid catabolic processes, fatty acid catabolic processes. So we went to our flies from the same generation, and then that we used them for RNA-seq, and we measured lipid content. And then we see that in males, we don't observe any reduction in lipid content after 7 generations, but after 31 generations, we observe significant decrease in lipid content. So we kind of see that the genes that continuously decrease or increase their expression, and so they are involved in lipid catabolism, for example, in these flies. And when we look at females, we do observe this decrease, but this decrease is actually not significant. But I'm thinking that it might be because of the assay that we use, because we use a colorimetric assay, and they may not really be that sensitive. So if we do some metabolomics, I think that we would be able to have a better resolution, or maybe it would be more sensitive, and we would be able to identify some reduction here as well. This is triglyceride, the total triglyceride, total lipid content of the flies. And this is whole body. And so initially I was agnostic to a group of genes that we call them the reverse complete or incomplete, because I thought it doesn't really make sense. But more and more, I start to like them a little bit more. And those are the groups that after a significant increase or decrease in their expression, they go back to the baseline expression. Some of them, they completely go back to the baseline expression, meaning that there is no significant difference at generation 31 compared to the founder population. Some of them, they are still significant, but there is a change in the direction. So these are the 5 classes of genes that we can identify in these differentially expressed genes, depending on the patterns of their expression. And so we measured some traits, including fecundity and this gene expression. And we also have other traits like protein, lipids, body weight. And since we think, or I think, that trait optimum might have been reached at generation 7, I was wondering what is the rate of phenotypic change in the early phase of adaptation in the first 7 generations versus the later 23 generations. And for this, to make the comparison more homogeneous or across all the replicates, we decided to use Haldane as a rate of phenotypic evolution, which I think is d over y. So d is the average, small d divided by sp. So d is the average between the phenotype, average difference between the average of phenotypes at 2 different time points. And these are log transform across different traits, and this one is SP is basically the pooled standard deviation. And yeah, so here I'll walk you through it. So in the first one, when we have generation 7, this is the rate of phenotypic change between generation 0 and 7. And the second one here is the rate of phenotypic change between generation 7 and 31. And if we expect that, or if I'm right, that majority of phenotypic change or adaptation has occurred early on, so we should observe a high phenotypic change in the first 7 generations and much smaller in the later 23 generations. And this is what we see. It varies among different traits. So one of the largest traits that shows the most change is the fecundity. So this is the fecundity in the first 7 generations and in the next 23 generations. I think this is the amount of lipid content in females and males, and this is body weight in dry body weight in females and in males. So we observe that there are different traits have different rates of phenotypic change, but the most, and we also look at gene expression, rate of gene expression change. And so these are the 5 categories that I already introduced to you, whether those that have plateaued, whether those that continuously increase or decrease in their expression, and those that basically change the direction of their expression. And we see that gene expression is among the traits that changes the most after fecundity, probably. And the genes, groups that belong to the plateauing ones, so genes that plateaued in their expression, and those that are still continuously decreasing or increasing in their expression, and those that they have changed their direction, so basically they reverse, they have the most change in the first 7 generations compared to the other groups. But basically all of them, for all of them, majority of change occurs in the beginning, in the first 7 generations, and not much afterwards. So since we already observed that the majority of adaptation might have already happened, so I was wondering whether this initial phase of adaptation has been facilitated by contribution of large effect alleles, and later on, in later time points, we have a contribution of small effect alleles. Basically what, Fisher and Orr talked about, and also what I think Sam talked about it extensively yesterday. But in this data set, we do not really have the fitness effects. What we could do is that we could use measure of pleiotropy as a proxy of effect size. And this is what Sam also talked about yesterday. So empirical studies show that degree of pleiotropy, meaning the number of traits that a gene affect, is correlates with the effect size. So the more pleiotropic you are, the larger effect size is. And that would mean that if you look for genes that are more pleiotropic, we could actually talk about larger effect size. And for this data set, although this is gene expression, we could estimate pleiotropy. And so we use two measures of pleiotropy. I think that axes are not readable. So we first use connectivity, network connectivity as a measure of pleiotropy. And you can imagine in a network, genes are connected to, in a gene expression network, genes are connected to each other. A gene that is highly pleiotropic in a node is connected to more nodes. So this expression affects more genes. So it has more edges compared to those that are less pleiotropic and affect expression of fewer genes. So we use already publicly available data set for Drosophila to estimate the number of edges or the degree of pleiotropy for each of those genes. And we can also use tissue specificity, meaning that those genes that are more pleiotropic, they're expressed in more tissues because they probably affect more traits. So, and then when we look at this data, these two measures, we see that the more pleiotropic our genes are, the less differentially expressed. And this is the same in either of these measurements. Either we use degree of pleiotropy as connectivity or tissue specificity. And when we bin this, depending on different bin sciences, we observe this decreases also pretty well. But when we look at our different differentially expressed genes where they have different patterns of expression, we observe this actually much better. So for genes that responded early on and then they plateaued, or they're continuously increasing or decreasing in expression, meaning that the expression in the evolved populations are modified compared to the founder population, which is basically this blue and green one, they're significantly less pleiotropic compared to the background genes, meaning that all the genes that have pleiotropic information. And when we look at those genes that after an initial increase or decrease in expression, they reverted to the baseline, we observe that these are the highly pleiotropic ones. So these are the level of pleiotropy is significantly higher than the background. And this is based on gene connectivity. And when we also look at tissue specificity, we observe this pattern actually pretty even slightly better. So I think these results show that, let me see. Yeah, so these results kind of show that adaptation is fast. And in the early phase of adaptation, large effect alleles or the more pleiotropic alleles have responded, but then they later on change their expression to the baseline level. But those that are less pleiotropic, they continuously increase in expression, or they basically stabilize at a particular gene expression.
Hello. Okay, now it's on. Could you go back to the previous one? So when you say that highly pleiotropic genes show less change in expression,
this is log 10 of connectivity. So these highly pleiotropic ones are here, and then their expression. And this, and the y-axis is the log 2-fold change of expression. So if the expression is higher, so you are less pleiotropic, and then the highly pleiotropic genes have smaller fold change.
And this is from 0 to 31, not just after it plateaus.
Yes.
So the change is from overall change.
0 to 31, yes, the overall change.
And then afterwards, you said that you see that it goes, there are some that goes back, like they have the reverse. Are those, like I didn't understand this bit, like
No, here, we, for the genes that we identified as differentially expressed, we categorize them into different categories depending on the patterns of their expression. So the plateauing ones were the ones that increased in, or modified, have the expression modified, but later on plateaued between generation 7 and 31. And those are the ones that have lower pleiotropy, but for those that after an initial increase or decrease in expression, they go back to the baseline level, these are the ones that are more pleiotropic compared to the background. And another parameter that we can actually use to find these patterns of adaptation is parallelism, level of parallelism. And basically, this is similarity between two independent replicate populations. So you don't really need to look at the green ones. These are the under sweep simulations, but for the purple ones or the red ones, so the dark one from a large population, and then the pinkish one is patterns of parallelism in a small population. So in the beginning, when the population is changing in basically in its mean of phenotype, mean of traits, we have an increase in parallelism, so replicates becoming more similar. But as soon as the population is at optimum, then its parallelism decreases. And we can also look at parallelism in our gene expression data. And for this, we use Jacquard index, which is simply the index of similarity based on the number of shared items between a pair of data set divided by the total number of the sum of items in these two data sets. And it can range between 0 to 1, 0 meaning that there is nothing shared between these two data sets, 1 means that everything is the same. And we see that in these large populations, at generation 7 and 31, there's basically no difference in parallelism, so it kind of has plateaued. And it's quite high, it's around 0.7, meaning that if you randomly choose 2 replicates, there's a chance that 70% of differentially expressed genes are the same. So in this part, I showed that adaptation to a selection regime that has multiple selection factors, including a new diet, adaptation to temperature, or early fecundity, could be fast and facilitated by changes in expression of many genes. We see that optimal phenotype could be reached quite early as 7 generations. We see just genes with strong change in their expression, they later on plateau, and these are less pleiotropic ones. And the more pleiotropic ones, after initial change, they revert to the baseline. Yes, and adaptation is highly parallel. But I also mentioned that we have selection experiment in small populations. So in these small populations, everything is the same except for the population size. The selection experiment, the environment, everything is the same, and the founder population. So here we're looking again at fecundity or fitness, and in these small populations are in this light pink. And you see that they have generation 7, 31, and 60, and we do not observe a plateau, but I will later on show our gene expression data, but I think that what is happening here, that they might also have been at an optimal phenotype as early as generation 31, but because of the small population size, drift really disturbs these populations. So that's why basically their selection, it fluctuates, and then it disturbs the plateau of all these traits or phenotypes, including the one that we have measured here. And when we look at gene expression data, again, the orange ones are the founder population, and the green ones are the evolved ones. Light green is at generation 7, and dark green is at generation 31. So we do see that there's some heterogeneity at generation 7, but at generation 31, most of them cluster basically together, quite separate from the founder population. And when we look at this correlation plot, which shows the correlation of gene expression and in populations at generation 7 and 31, we see that there are still some genes that have shared gene expression pattern, around 55% of the genes. But when we compare them to the large populations, I'm showing here, so the correlation is actually less. We see that here a majority of genes, around 75%, are shared, but here we do not observe this pattern, and that might be an indication that these populations are, of course, not as efficient in their adaptation, and the potential optimal phenotype has not been reached at generation 31 here. And we also can classify our genes, differential expressed genes, in the small populations, depending on the patterns of expression. We still observe all these patterns, whether they plateau, whether they still have this monotonic expression change, or whether there is, basically they have reverted. But the fractions are much different. So the plateau fraction, which I'm showing it here now with comparison to the large population, as fewer number of genes have plateaued in their expression, and majority, or a large fraction of them, still continuously changing their expression. Again, suggesting that, yeah, these large populations have been much faster in their adaptive response. And when we look at patterns of repeatability, of course, this is what I think I did expect, that it would be more heterogeneous in the smaller population. So we see that at generation 7, the patterns of heterogeneity is quite, patterns of repeatability is quite heterogeneous, and is smaller, less than large populations, but it quite increases at generation 31, but it still is less than what we observe in our large populations. So initially, when I was, when we analyzing this data, I thought that small populations might, because of strong effect of drift, some alleles may not really be able to respond. So they may sit in a different fitness optimum, just because drift really disturbs a lot of allele frequencies. But maybe after this data set that we have, and this analysis of gene expression data, I think we probably are sitting more or less at the same optimum, but it's just that the small populations, the patterns of phenotypic change is less efficient, but also phenotype is more disturbed because of drift around this fitness optimum. It's exactly what I think Luis Miguel also showed in his model. So is there any questions so far? So in this part of my talk, I also talked a lot about trait optimum, but I think it is also clear to everyone that we still do not know what a selected trait is. We just know that when it might have been reached in our large populations. But you can imagine that in our large populations, when we expose them to this high protein diet, if you can imagine, this has been the initial phenotype of the population, and it's like a fitness function, and then there was some shift, and this was a new trait fitness function, and this was adaptation to a high protein diet plus temperature adaptation and early reproduction. How can we manipulate this and make it less or more, this distance to this new trait optimum? So what we could do is that we can keep temperature adaptation and early reproduction constant and basically do not change the diet. If we keep them in the ancestral diet, it would be hand waving, as if the shift of this to this basically distance to this new trait optimum was actually less. I think this also, I think Luis Miguel showed in some of the studies, they changed the intensity of selection regime just by adding more stressors. So if we assume that we could play around with this combination of selection pressure, selection stressors, we could actually study the effect of selection pressure or distance to this new trait optimum on the adaptive processes. So we did another experiment alongside this one that we performed. So I showed the results of patterns of adaptation in these populations, small populations that were exposed to this high protein diet. So at the same time, we also started some evolution experiments in small populations, but we did not expose them to this new protein diet, and we maintained them in the ancestral diet, which is a protein poor or carbohydrate-rich diet. So you can imagine that these populations now still have to make this shift, but this shift is actually much less strong than the other populations. And these are the patterns of phenotypic change in these populations. These are the orange ones, and I'm showing them next to the previous treatments that I showed you. So here you see that, again, we have this dramatic increase in fitness or in fecundity, but it kind of plateaus pretty nicely, as I would say nicely as our large populations, as early as 7 generations. So it basically means that what really disturbed the patterns of this phenotypic change in our small population that we're adapting to this high protein diet is not necessarily because of small population size, but also the selection pressure or how much they had to change phenotypically to be in this, to reach the optimal phenotype in this new environment also plays an important role. And again, when we look at gene expression, the orange ones, again, are the founder population, and different shades of cyan or navy blue are at generation 7 and 31 in these small populations that are being maintained in the ancestral population, ancestral diet. And we see that there is some nice differentiation, and with PC3, we could actually nicely separate generation 7 from generation 31, exactly with the same pattern that we can actually do in our large populations. And when we look at the correlation of gene expression fold change after 7 or 31 generations compared to our founder population, we observe this nice correlation, and almost 75% of genes are shared, which is basically means that the magnitude of change at generation 7 and 31 is highly parallel. But we have around 11 or 14% genes that are specific to each of those time points. And when we look at different patterns of co-expression, question.
Sorry, when you say shared, it's shared with the other treatment, or is shared?
No, it is shared between at generation 7 and 31, meaning that if we compare gene expression at generation 7 to our founder population and at generation 31 to our founder population, we see that 75% of differential expressed genes are shared between these two, but also I think the magnitude and direction of change are also highly correlated. I think the correlation of this one is around 0.92 or so, if I'm not mistaken.
Okay, and you probably will tell us later how much is shared with the other treatment.
It's in additional slides, but yeah, I can show you. It's a lot, yes, but I can show you. And looking at the patterns of co-expression of different groups, the ones that plateau, the ones that are monotonic, or the ones that have reversed, we observe that still majority of them plateau, and a small fraction of them still continuing to decrease or increase in their expression. And it is more similar to what we observe in our large populations compared to the small population. And when we look at patterns of parallelism, we do observe something between the small and large population. So these are these dark blue colors. So at generation 7, level of parallelism is less than generation 31, but it is much, parallelism is much higher compared to the small population on high protein diets compared to, compared to those. And so here in this PC, I'm showing the founder population and all these 3 treatments that I introduced to you at generation 31. So the large populations in this high protein diet, as in this purple color, the small populations on the high protein diet are in this green squares, and the ones that are smaller population on this ancestral diet are the blue ones. So you see that there's a clear clustering between founder population and these evolved ones, but we cannot really distinguish any of these either at PC2 or any other PCs. And I think that kind of suggests that all these populations, despite some of them share the diet and some of them do not, they all are moving towards the same optimal phenotype in this new environment, and that is why we observe this overlap in gene expression. I don't know if it's because we have standardized them, so to put them mean at 0 and standard deviation to 1. I don't know if it is because of this but you are right. So you mean here we have this parallel response, but here there's a shift. I mean, this reverse.
I mean, another way to present them concurrently, especially comparing the two ends, is just to present a distribution of how much the rank changes across this.
How much there?
Just give a histogram of what the rank is, the correlation among ranks, because that'll give you a much better idea than, because eyeballing here, you can't tell what fraction. Pardon me?
For each of these groups, you mean?
Yeah, so if you basically take the idea about looking at plateaus, so take the endpoints, rank the transcripts, look at a correlation among the ranks, and present that as a histogram as well. That'll allow someone like Mike to look for how many crossovers you're seeing.
For different treatments, or
It sounds like the point you're trying to make is that well, I mean, within each model, you can see when they plateau, how often are the ranks the same, even though the general level is across, but how often do the ranks differ? Just presenting that in addition to this would give more information from it.
so. So in this part of the talk, I showed we could identify when populations are at an optimal phenotype in a new environment, even though we do not know what the actual selected trait is. But I think for a lot of cool quantitative genetic studies, we really should know what our selected trait is to basically correlate them with the frequency of adaptive alleles. And so, although we have measured fecundity, we look at gene expression, we look at lipid content or resting metabolic rate, but none of them could actually be the truly selected target. And with all the models that, for example, yesterday and today, especially, we've looked, we always need to know the trait, and we should basically, based on that trait or the variance in that trait, we measure fitness. So I've always wanted to see how we could perform an experiment that truly mimics the assumptions of these theoretical models. And I think for one to be able to do such an experiment is that we should be able to, in a high throughput fashion, phenotype our population to know the distribution and somehow convert that distribution to fitness function, similar to what Louis Miguel suggested. And then we would be able to perform a selection experiment where we gradually shift our trait optimum, and after that we could do stabilizing selection or all sorts of things. But that requires a high throughput phenotyping that would allow this experiment to actually be happening in multiple replicates. So along that experiments that we have been doing, we were also on a hunt of a high throughput phenotyping method. And I think we might have found one. And we measure, that is measuring the size of Drosophila egg or Drosophila embryo using a high large particle flow cytometry. So you all know how large flow cytometry works, which basically is a flow cytometer. Flow cell at small objects, normally up to 200 micrometer, pass through this, and then normally they are labeled, and then we can actually separate them based on the fluorescence. But now there's a new batch of our flow cytometers that are called large particle ones, which can accommodate objects of up to 1,500 micrometer. And that is like Daphnia, Arabidopsis seeds, Drosophila eggs and larvae, up to I think 13 star, and a lot of other things, as long as they are not bigger than 1,500 micrometer. And the way it works is that as an object passes through a flow cytometer, it blocks light. And then the duration of time it blocks the light corresponds to its size. So larger objects block light for a longer period of time, and smaller ones for a shorter period of time. So you can actually use this time of flight, the time that a laser beam is blocked, as a proxy of size. You can actually have reference speeds to measure this properly. And after that, you can very accurately and quickly phenotype the whole population. And what is cool about it is that you can actually specify particular size beams, however you want, and choose objects of a particular size. And that means that you can perform the selection experiment that I just talked about, here. So as a proof of principle to see whether it works, we did a very small scale study, and we measured egg size in an outbred population. And this green one is kind of like a the distribution of fitness, trait distribution of embryo size. And although we should have a fitness function that is actually much wider than our trait distribution, but just to see whether it works, we used the distribution of traits as a proxy of fitness function. And we decided to have basically the same, use the standard deviation of distribution of trait as the standard deviation of new fitness function, and we moved the trait optimum one standard deviation. And that means that we wanted the average egg size from 440 micrometer to now be 540. And the way we do this selection experiment is that we phenotype the founder population. We have this fitness function that is fixed throughout the whole experiment and does not move. And then we choose the embryos that fall under our new fitness function. After 10 generations, our fitness function does not move, but the distribution of our trait, our egg size, have shifted towards our new mean. So then again, we choose the individuals or the embryos, eggs that fall into under the new fitness function. And we continue with this, and eventually they would be really close. And after that, then we can basically perform a stabilizing selection. So we did such an experiment, and we selected the same procedure that I explained for around 27, 30 generations, and we already see a response. So the green curves show the distribution of egg size in the control lines where we do not select for any particular size. We just run them through the machine. So these are our control ones. And we have two selected lines where we select based on this to have larger embryo size. And we observed that the average embryo size is significantly higher in these selected ones. And to see whether, because we expect that if flies need to lay larger eggs, so other traits related to egg reproduction should also be modified. So we looked at fecundity, the number of eggs that flies lay, and then we see that for those replicates that are selected to actually have larger eggs, they lay fewer eggs, and for those that have normal size eggs, so this higher. So it's basically the total energy investment is the same, but it's just either you have bigger eggs and you lay fewer of them, or you have smaller ones and more of them. And also when we look at body size, body weight, in our females, we observe that these are significantly heavier compared to the control ones. And although our traits should not really have anything to do with males, because we're selecting for a trait that is specifically controlled by females, but we also observe an increase in body size in our males. And I think just because the genetic correlation is just pretty high between these two. And no, these are adult. These are adult females and adult males. Oh, this is weight. This is body weight. Body, yeah, sorry, yeah. We not when there are eggs, but when we let them hatch, of course, we know whether they're males or females. And so I'm quite excited about this one, this type of phenotyping or selection experiment, because it really allows you to perform selection experiments that we can test many of the models that we heard about yesterday and today. And that would mean that one of the simplest thing is that you can shift the trait optimum and see how it affects the alleles that respond to adaptation. Whether you have larger fraction of large effect alleles where the trait optimum is further, or you basically have more of a small effect ones. But you can also play around with the width of fitness function to make it make the selection stronger or less strong, Or you can also, when the population reaches this new trait optimum, when you want to perform this stabilizing selection, you can either do symmetric stabilizing selection, or you can actually modify some of those that, for example, Luis Miguel showed, that some of the stabilizing selections were not really symmetric, actually. So these are the types of experiments that we are planning to do using this method. And if you have any suggestions, we would be extremely happy to hear. Yes.
The selection you imposed was very strong because the width of the fitness peak was as high. So whereas Nick was saying that we usually think like the ratio, let's say the ratio of the width of the fitness peak to the phenotype distribution, if you square that, it would be around 20 in the models. But I'm wondering if this is why you got all this correlated response by these other traits, because you might have more effects of linkage in this case if you impose such strong selection, building up LD with I don't know, things related to male body size, you were saying? You got this correlated response?
But I think for body size, males and females are highly correlated.
Yeah, but you select for X size, right?
We select for X size and...
No, but okay, this may be a true correlation, but this might be interesting if you have weaker stabilizing selection, because this is also a strong directional selection then, and it might be that you would see one of these things you could look at is whether you find fewer correlated responses of other traits. And something else I wanted to point out
on the width of our fitness function. You mean?
Yeah, and something else I wanted to point out when you mentioned there was this trade-off between egg number and size, right?
Yes.
Some people I know in Australia, they just love burning eggs. And they count the total energy, apparently, that you get in these, because an additional trade-off is how much resources they put in the total number of eggs. So I don't know if this would be a correlated trade, but apparently it's easy to do when you just burn them. But then you cannot use them to do the next generation.
But you are right about this strong selection.
Yeah, we will have to do something like this.
Yeah, you're correct about this. And I think I just have to mention that this was only a preliminary analysis with few replicas to actually see whether it works. And I think, yeah, we should definitely think of the strengths of selection for the actual experiment. Yeah. Any more questions? And I think with this, I would like to thank a lot of people, especially Christian, a lot of people that contributed to the first study, the thermal adaptation study, my collaborators, and also most of these analysis is done by my super PhD student, Claudia, for sending me plots even last night at 1 o'clock. So thank you.
I'm a bit confused by your correlation between 7 generation and 31 generation. Why would you expect that it remains correlated? What is it based on? Because, you know, it could be that the first step is simply one large effect transcription factor that has changed, and then maybe if you have smaller population size, it starts a little bit later.
So you mean why they're correlated?
Why do you use this as an indication of replication when in practice it's just a little bit random what's going to fix first? Or I mean, for me, it's if you have always the same transcription factor that goes up at the beginning, then you're going to have this all the time. It depends a little bit on the genetic basis of the first step.
You mean why we use it as a proxy of parallelism?
Yeah.
You are right that
you inflate a little bit your perception of parallelism, I think.
It should be used at the genetic level, because then what really matters is that what are the alleles that are selected and whether these are shared. So we don't have genome, I mean, the student who's going to analyze it is going to start in September, but why I was interested in looking at it is that the data set, the temperature adaptation data set that I showed, and we observed large heterogeneity at the genomic level. Christian students, when they look at it at the gene expression and at the metabolomic level, they also observe highly heterogeneous response. Even the heterogeneity is actually much more than the genomic level. And that's why I was interested to look at this, but it seems to be pretty high. But you're right that we should really look at the parallelism at the selected alleles. not necessarily gene expression. But I think that may suggest that beneficial alleles in these replicates might also be shared, but to be really sure about this, we have to really analyze the genomic data.
So do you think the convergence that you saw, or increased convergence in the later generations?
Which convergence?
Well, you saw more parallelism at later generations, yeah?
For our
these
small populations, yeah.
Small populations, yeah. I got confused, maybe the normal food.
Yeah.
So is this what you expect? Or does it mean that basically you have actually less redundancy than you would, than the generation 8 suggests, that you basically have taken a path that is increasing fitness, but there are so many high costs that you basically then converge to another solution? Or what is your interpretation of it?
In my opinion, it's mostly technical, because we do observe high correlation in the gene expression in these 2 time points for all of these treatments. So it basically means that genes that are differentially expressed at generation 7, they're also differentially expressed at generation 31. But sometimes, because the magnitude of change is not high at generation 7, we do not necessarily identify them there, but we identify them at a later time point. And when we do this Jacquard index, we perform gene expression for each replicate separately. We have less power, but it doesn't mean that they are not they have not modified their expression in the same direction. It's just we do not observe, find them significant. So in my opinion, it's probably all technical, but because I can't imagine why redundancy would affect, why it would affect it in later time points.
You could think of big effect alleles being important in the beginning, and
then they're lost, and that is why we lose.
And then the other ones take over. I don't know.
But it becomes more parallel later on.
So. Yeah, exactly. So
I would
individual replicates, depending on drift, you have one big effect going up in one replicate, not in the other. That's how you generate the heterogeneity. But later on, you get rid of these big effects and take over
the small
effects,
ones.
and then you get the convergence. But maybe your explanation is better. It's simpler.
We are sequencing generation 61 and for more replicates, and I think then I would be able to talk, explain maybe better with more data sets. Yeah.
So you were showing this trade-off with the number of eggs. Do you think it's going to limit your response at some point because you're going to have too few eggs, and then?
Which one? Sorry.
You were showing, unless I misunderstood, that when you select for larger eggs, they produce less.
Yeah.
So do you think you're going to, because it's sort of a well-known trade-off that is sort of maybe a hard limit. Do you think you're going to limit your response at some point? And
that population size will shrink.
I think at some point the ones with the bigger eggs, they would produce too few eggs, and then they will die.
That is a possibility, but it has not really happened to us yet. We see that majority of, we can maintain our populations with a adult population size of 1,200, and we always get around 2,000 eggs to set up our next population. So despite this reduction in fecundity, I think we have not really observed, we haven't observed this pattern. And yeah, and it's possible that, like the other data set that I talked about before, there might be an increase in fecundity anyway, because we're selecting for early fecundity. So even maybe despite the fact that they're producing fewer eggs, but since their fecundity is higher compared to the previous populations, then it may not really have an effect. But yeah, we have not observed this, but yeah, it's something to consider, I guess. Because initially I was afraid that maybe females, because they have larger body size, maybe larger eggs would turn into females. So eventually we would not really have any males in our population. But it seems that we get, I don't know, 50-50%. So
Yeah, because compared to selecting on temperature, where they probably have to modify their metabolites, and so where here you might reach a limit where it doesn't respond to selection just because of the trade-off.
True. Yes, yeah, but I think that should be same for all the truncating selection experiments, because you don't know the response to selection really depends on the genetic basis, and for most of the cases we don't know how many alleles are really responding or contributing to this allele. Yeah, but it's something that I think we should be really careful. Okay, thank you.